So hi, hello everyone again. Welcome. I'm Oliver, Microbehunter here. And yeah, basically in the background you can already see the pond where collected some algae. Let me just get a few of the things working. Okay, I think everything seems to be working. I hope that you're able to hear me as well. And let me quickly check here. Okay, everything seems to be going fine. I hope so. Okay. Yeah, okay. Here I am again. And it's been a hot day. It's not only been a hot day, it's been a hot week. Here in Europe, where we are right now, we're experiencing one of the hottest days ever on record, and that's good news for the algae. So algae growth has been exploding, therefore. And it was just yesterday when I basically went to a local pond. For those of you who are in Austria, where I am, this is the university pond from the University of Linz, JKU. And it's basically where I always go to collect some water samples. There are plenty of ducks there. This basically means that, of course, they're producing waste, which is a good fertilizer for the algae. I think there are also some fish in there. And the pond water looks extremely green, obviously. During winter time, it's much more clear, the pond water, and you can see actually over the seasons how the color actually changes. Yeah. It's not the first time, obviously, that I've been there. I've just a couple of weeks ago, I've also collected some samples, but every time when I collect samples, it's a little bit different. But I have to say that this time the pond is extremely green. I've had to laugh a little bit because right at the beginning of the chat, at the very beginning, yeah. It was the second chat message from the top is, did you get the algae from the reflecting pool? I had to laugh a little bit because actually it's the whole news report on the reflecting pool that actually kind of tipped me off, and that's why I actually checked out this pond over there. For those of you who don't know, they're currently in the media in Washington D.C. They've been did some maintenance on the reflecting pool, but unfortunately the algae started to explode there. And I basically said, so what do you expect? You've got some standing water somewhere, you've got light, you've got nutrients. Of course, algae are going to grow. I mean, it's obvious, right? It's a pond, it's a natural environment. Yeah, nature is hard to come by. And if the algae have enough sunlight and nutrients, then of course they're going to start growing. And because it's been so hot in the last couple of days, this basically contributed to that. And I have therefore, what I will do today, therefore, just a little bit of an overview. I did prepare already a little bit. I did some centrifuging as well, simply so that it's a little bit faster. And I do want to try out something as well. I want to try, of course, making again a couple of permanently mounted slides. And I would like to, use test some mounting media. So I've got glycerin gelatin. Many of you already know that. And I'm going to be also testing a new type of glue. Of course, the green screen, therefore you're not able to see that. But there's a new type of glue that I would like to try out. And we're going to see whether that actually also works. And then later on towards the second half or towards the end of this live stream, I want to go back a little bit to the insect wings because I would like to quickly also show you there how we are basically how the slides from last week's how they are. Okay, so if you want to write comments, please feel free. I will always be watching the comments a little bit with one eye. And let me quickly do another thing. I did something else quickly. Let me quickly check because I did also get an email, and I need to quickly check something here because I would like to also share, if possible, later on. I got also some pictures to share with you, and I quickly need to check if this also works. So I'm basically logging in right now, and okay, I got those as well. So what we're doing here, we are again. Okay. Yeah. Very quickly, just to show you what I've been doing. So this is basically the sample, and a little bit something that is always a little surprising is that actually once you take it out, it does not look quite as green as when it's in the pond. Of course, this has to do with the fact that the pond is much deeper. But when you take it out, it's actually much more transparent. But still, the density of algae is, I think, still a little bit low. What I've done is I've been centrifuging it. So what I'm going to do now is simply to compare this. So I've been centrifuging it about 5 or 6 times. Right, so you see that it's very, very dense here. So what I would like to do now is I simply want to take the original sample under the microscope, and we're going to see that actually there are not so many algae visible, right? Because the concentration is simply too low. Yeah, so that's standard procedure. You put a drop on here, cover glass goes on top, and yeah, I'm just gonna forewarn you, it's not gonna be super exciting. Okay. But I always do that at the beginning in any case, simply to get a better overview of what's there. Let me see, this is kind of interesting. Okay, no, let's have a look here. And I'm going to switch the scope view, and this is what we see. Okay, okay, here we go. So I'm this, let's focus this a little bit. Yeah, and yeah, we kind of go up with the magnification here. Yeah. And we can already see some algae here. What's this dark thing in the background? Yeah, there's some dirt on the microscope slide. It's not to be bothered. And we can see also some ciliates or flagellates, and they're still moving around. Yeah, so we go up a little bit with the magnification. What I'll be doing now is the following, is because I'm using now DIC. I'm not seeing the natural color, so I will remove that and we'll just go regular bright field. Right, and then we can actually see better that it's actually a little bit greenish, and this allows me to actually identify the algae a little bit better. Yeah. The thing is the following. The question is are we centrifuging the slides to get more? Yes. I just wanted to show you first the non-centrifuged sample as a reference. Okay. And if you just basically go around here, I mean, lots of debris, dust and dirt, and I don't know, and occasionally some algae. I also want to go up a little bit with the magnification later on to do a little bit of oil immersion. Yeah. And very often, if you look at this now, very often we see that there are some of these small colonies where the cells are in fours, like over here. There are 4 of them next to each other. That's also something that's quite common. Yeah, but I think we all get the idea a little bit that they're not that the concentration is not that high, as expected, obviously, as expected. Ah, look at this. What? I bumped it here. What is this here? Let's follow it around a little bit. It's also green, also photosynthetic. But they're way too fast. Yeah. Yeah, and essentially what I like doing, I just basically put a specimen on the sample under the microscope and I look around hoping to find something more or less interesting. Yeah. Yeah. So that's the thing. I'm a little bit confused now. Why is it always in 4S? What does this mean? Okay. Okay. So we have yeah. So this looks a little bit yeah, or filamentous algae. Oh, 4S in force. So okay, sorry, my mistake. I've been thinking for yeah. In force. Why is it always in force? That's it's not always, but many of them are actually are. You see that there are, of course, other microcolonies as well. But the algae with the genus Senedesmus, that was actually also one that was reported in the media as having been found in the reflecting pool, is actually in force. And this, yeah, probably has to do is because as it divides, one cell divides into 2, and then each one divides again, and then you have 4. But that's actually only one possibility. But I think you get the idea that yeah, it's kind of how do you say, yeah, not very dense. And indeed, there are yeah, it's the last comment over here. Sometimes they're embedded in a matrix. That is correct. Okay. There are sometimes, like for example, those here. Yeah. You can very often see, but the matrix is sometimes difficult to see. Let's open this up a little bit. But very often when the cells are sticking together, like this, then sometimes they are embedded in some kind of a gel-like matrix. You see the long filamentous algae on the bottom. That's probably some kind of it's a cyanobacterium, right? And you see that those cells here, yeah, the round ones actually they are sticking together in a gel. Yeah. Can it be made visible? I guess with staining, maybe. I've not done that yet, but usually always when you add some kind of a stain, you also end up somehow destroying the cells a little bit. I suppose it's some kind of very often those gel-like matrices are made of something like a polysaccharide or something. Yeah. So then you see over here also some diatom, but not diatom, cyanobacteria. Yeah. Yeah, diatoms, again, I've not seen so many. Diatoms, sometimes they settle to the bottom of the pond, and I've not collected now samples from the bottom, but actually so-called planktonic samples which are floating around. But yeah, you see, look, do you see the spiral-shaped bacterium there as well? This is spiral-shaped bacterium right to the right of the. Let me quickly. Yeah, it's difficult to see. Let me quickly switch over to phase contrast, hoping that we're able to find it there. I don't know. Not so much visible. It's not phase contrast. Doesn't matter. Okay, let's stick to the algae. They're interesting enough. Okay, so what we're going to do now is the following. I want to now have a look at the centrifuge sample because I do want to make a couple of permanently mounted slides as well. So this basically means I'll be looking here in closer detail of what we got here. Okay, so let's. So you see over here, this is called the pellet. So what I've done about 6-7 times, I've filled up the tube with this pond water, and I was spinning it very quickly. And I decanted, I kind of threw away the supernatant because all of the solid particles were pulled towards the bottom here. And look, the supernatant looks pretty clear. Just compare this now a little bit, right? This is basically the, and compare this to the original. Huge, huge color difference, yeah. So basically that's this one here, that's waste, so to say, and the algae, the solid algae, are now basically here on the bottom. And what I have to do now is I have to resuspend it. So what I'll be doing is I'll be taking some of the liquid from the pond water, and I'm going to put it in here, and I'm going to just mix it. And I'm going to try to resuspend everything completely. So this is a really very high density of algae, probably even a little too much, maybe. Okay. But at least this will then give us a much easier time actually observing under the microscope. Okay, just mix it a little bit like this. And because it is so extremely dense, I'm just going to take a very small amount here. I'll put it on this microscope slide. I need to grab myself a slide from over there. So here we go. A small drop. Here it is. It's already very dense. And the rest goes back here. Cover glass goes on top, and this will now spread out the algae quite a lot. Here we go. Okay, and now let's have a look. So I'm putting it in here. The scope view, so that's still the old one, the original sample. It's still the original sample, and let's move over. And that is now the sample. Nice. That's now the sample after I centrifuged it. And I think we can all agree, let's go up with the magnification. I think we can all agree that this actually looks quite a bit more interesting. Okay. A whole bunch of, basically a whole bunch of not diatoms again, cyanobacteria. And yeah, this one beautiful, I think. Yeah. Let's go up a little bit with the light intensity. Yeah. And we're just going to simply explore this a little bit. And you know what I'm going to do? I think I'm just going to also then later go up with the magnification yet further I'm now using my 60 times objective, but then I'm going to use oil immersion 100 times. And if you just look around, then you can see, and we'll be watching for out for those. Let me turn on the arrow. See those here where there are 4 of them next to each other, and there are those tiny little extensions here. That is very often Cenedesmus or Desmodesmus, and that's apparently also the one that they found in the reflecting pool. Honestly, not surprising because they are so extremely common. Here again, yeah, in force, yeah. So quite nice to see a little bit the biodiversity, and then what you can do if you want to, you can simply observe the change in the algal composition over the months. Yeah, yeah. So I'm just occasionally also reading a little bit some of the comments here. There's some discussion on the structure of the universe. Look at this here. That is moving around. It's quite fascinating. What I'm going to do now is, let's have a look using different contrasting techniques. I don't know. We're just going to leave it here as this. Okay. Haha. Stenodesmus. Yeah. There are several different species of Cenedesmus, and because you are referencing Das Leben im Wassertropfen, this is basically a book that I recommend, and believe it or not, look what I got here. Okay, here we go. This book is a reading recommendation. No, not a reading recommendation. It's a reference book, identification book. Yes, it is written in German. And no, you do not need German in order to actually understand it. Yeah. The reason is because it's mostly drawing based. Now, I am a little bit afraid of actually showing you the contents of this book for copyright reasons and so on. But I know that you, at least the introductory chapter, you can get online. You can read online on Amazon. But the main part with all of the beautiful drawings, basically, unfortunately, is not in the preview. I highly recommend this book, and so far I have found pretty much almost everything that you can find in a water sample I found in here. Ooh, I've seen something here. Something. Look. Looks a little bit like a heliozoan. No, but really, that's interesting. But heliozoans actually, why is it green? This one here with the spikes. Yeah, quite interesting. In any case, yeah. If now, however, there are also, I just also found a free reference which I would like to show you. Where is it? If let's do this here. No, you don't see that. Let me see. Where is it? This one over here. Are you able to see that? Yes. Okay. So this is actually a drawing from a book that I found online. Okay. And so I kind of recommend that. Maybe I can post the link. I'm gonna post. It's a PDF that you can download. I'm going to post the link now into the how do you call this? Into the chat. So let's see if this does not work. I have some keyboard. Oh, come on. What? Oh, gee. Occasionally my keyboard starts to act up a little bit. No, I cannot copy paste even anymore. Maybe the battery is dying. Oh yeah, now maybe I'm going to. Yes, here it is. Okay, I was able to manage to post the link, and there are some drawings in there as well. And as a start, why not try? Why not try this free? Why not try this free identification book first? Yeah. So you can actually see it's. And this one here in the center, number 26, that's the one that I've been talking about. And over here, it's also described. Yeah. You see it's flat colonies of 2, 4, 8 elongated cells. Yeah. So just as a little bit of a as a possible starter. right? You don't need expensive identification books. So let me go back to the scope view again. Let's try a different contrasting technique here. So that is not DIC again. You see, it's a little bit low in color. Yeah, here we go. Yeah. Okay. So, yeah, yeah, I know. Maybe I'm a little bit overly cautious because it's a German publishing company. Yeah. So, and it says somewhere that you're not allowed to reproduce it. Actually, using it for a live stream is not real reproduction anyway. But I'm a little bit extra careful. Yeah. But I kind of like yeah, simply exploring. Yeah. The water sample like this. And what I would like to do next is the helio. Where is it? Did I lose it again? This is what we were assuming to be a heliozone. You know what I'm gonna, and I'm not even sure if it is one. I thought I saw the spikes here. I'm gonna do now the following. I'm going to actually go oil immersion now. Okay. 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 So let's do the following. I'm going to go oil immersion now. So this means I have to use another oil immersion objective, which is of course way without oil. Not good. We have to center it, of course. And of course, without immersion oil or anything, it's going to be very difficult, but let's try to find the focus in any case. So this is now without immersion oil. I don't know if you're able to find it again. Doesn't matter, we're just, maybe it's the one here on top. Okay, but maybe it's a good idea to just remember how it looks like without immersion oil. Okay, without immersion oil. And yeah. Okay. Yeah, there's a question about my optics, which I will quickly explain after put the immersion oil on here. So I'm going to lower this here. I put immersion oil and then I won't be able to go back anymore. Okay, so that's also important. Once you've got immersion oil on this slide, it's, yeah, let's spread a little bit. And here we are, and as you can see, this is already much crisper. Yeah, so that's basically now, yeah, and just a second, this is also not right. I have to set the right, here, that's the way I wanted to have it. Yeah, so this is not a maximum magnification that I'm able to show you. Okay, and over here, this is now a bright field, and that is not DIC. And I will give you now a quick, answer to this question here. What objectives do you have on your turret, and are you using phase contrast objectives for DIC? No, what I've got is the following. I've got, you have to count them. 1, 2. Yeah, I've got the following. I've got these are basically so-called fluoride objectives, and this basically means they are semi-apochromatic objective. So they're not quite as expensive as the apochromatic ones, but nearly. And I've got a 4 times, a 10 times, a 20 times, a 40 times, a 60 times, and a 100 times objectives. These are regular fluoride objectives, so to say, and they can be used for DIC. Okay, and I have one phase contrast 40 times objective, and because the phase contrast objectives are different. Now, what I could do, and I'm not going to do that because it's expensive and it's not necessary. I could, of course, exchange all of those objectives with for phase contrast, but then for each objective I need to have a separate phase annulus. But then I would not be able to use DIC because DIC also needs its own prism. So you see, that's a little bit the problem. I cannot have everything, but I'm kind of with my compromise that I have. I'm reasonably happy. Yeah. So no, I'm not using phase contrast objectives for DIC. Yeah, these are separate ones. Yeah. Yeah. So those, yeah, and I'm not sure if it's a heliozone or not. Yeah, but yeah. Yeah, so that's the, yeah. So, but let's have a look a little bit. Let's do a little bit more exploration work here. Yeah. And then let's try to make a couple of permanently mounted slides. Yeah. With DIC, of course, as you know, you can adjust this a little bit. So maybe let's make it a little bit brighter. Yeah. So yeah, and simply some exploration. Usually, if you want to see, I don't know, like rotifers or ciliates or also more nematode worms, microanimals, in other words, you don't go directly into the pond water itself, but you actually have to go more into the sediment because this is where a lot of decomposition is happening. So those algae, they like to float towards the light, obviously, right? And they produce the oxygen there as well. But as soon as things start to decompose and decay, they sink down, and this is where you find totally different organisms. Yeah. Permanent slides with pond water, yes, but not liquid. Okay, so that's a difficult thing, and that's why I would like to try that. And what time is it? Oh yeah, it's almost half an hour now. So this is what I'm going to do next. I'm just going to leave it there right now. So every now and then we can always switch back to have a nice little look. Yeah. But I'm going to now go back to the table because we need a little bit of drying time as well. So first of all, making permanently mounted slides of pond water is difficult. Okay. The reason is the algae are delicate, and as soon as they dry, they basically start to shrink. Okay, so what we have to do is we have to use a mounting medium which is water-based, which basically, yeah, also, because if I use, for example, like last week I used Euperol for insect wings, it's perfectly fine. But if I use Euperol for algae, for example, it's going to dissolve the chlorophyll, the green pigment. Yeah, so that's not really good. So what I'm going to do the following is I'm going to take a small sample. This is very, very slimy. Yeah, so it's maybe even a little bit too dense. And I'm just going to spread it apart. Okay, of course, there is going to be some surface tension here. Yeah, and I'm going to allow it to air dry. And I'm just going to do it also with another one because I will be making... Do you know what? I mean, this is highly experimental anyway. So what I'm going to do is why not just simply make several of these samples on the same slide? Why not? I mean, this is a little bit of a comparison. I save some of the slides. Yeah. So, and the first thing that you have to do is you have to dry it a little bit. Okay, and this is usually the problem part, the drying, because this will actually cause a shrinking of many of the algae, right? And that's actually also one of the reasons why I would like to try it, because yeah, I want to see how it works. Okay, yeah. Now there's no space for labels. That's correct. I should have actually a pen. Damn, where did I put it? I should have a marker where at least I can label the, how do you say, the edges a little bit so that I don't remember, don't forget. Yeah. So this is basically something where we have to wait now. I mean, theoretically, I could use my coffee warmer. Yeah, or we just, I know it's actually already drying. It's so hot here that that essentially, yeah, maybe I don't even need to. Well, I need the coffee warmer later a little bit for melting this here. I have to melt that. Yeah, but I'll just give it a little bit of an air dry now. Let's see how well this works. The humidity is also quite high. Yeah, and let's go back here and let's see what we're able to find here. Yeah, you see that. Yeah, lots of stuff here. Ah, interesting. I don't know what that is. See, that also seems to be some kind of a colony. Yeah. And one of the nice things, again, about this is that depending on the season, depending on the conditions, weather conditions and so on, of course, you're going to get different specimens to observe. Yeah. Also, if you go to the bottom of the pond, this is where you, on the surface, decomposes, you're also able to find some amoeba. But here in the open water, so to say, of course, you've got mostly photosynthetic organisms. Okay. So that's the whole thing. Try one with PVA glue. That's, as a matter of fact, something I would like to try with PVA glue as well. And indeed, it's something that should work because it's both water-based. So, but I do not want to have too much water either. So what I'm going to do now is the following. I'm just going to give it a try. We're just going to, yeah, desk view. And what I'm going to do now is I rarely use this glue over here. Let's put this up a little bit. and different countries, they might have different ones. So this one here is, of course, it's labeled in German, and they have different versions of this. One is this one here is water-based, yeah, and I'll be trying this one. I assume it's also PVA based, yeah, because there's no reason otherwise. And I did not dilute this down at all. I'm just going to apply now a drop here. And the next week we're just going to see how successful or not successful it was. It actually, this is a huge drop. I think way too big. It doesn't matter. It doesn't matter. This is experimental. So cover glass goes on top. Let capillary action do the rest. And of course, it is quite likely possible that now, let's turn off the lights here. It's quite likely possible now that maybe some of those algae are also being pushed to the side. Can I not focus this? Here, that's better. Yeah. So that's number one. Number two is, of course, Elmer's PVA glue. Okay. And that's the one over here, which should be fairly similar. This one here, I diluted with a little bit of water, so it's a little bit more liquid. And let's also put a cover glass on top. I have to find a reasonably clean one, because I cleaned those cover glasses as well. Okay. Here we go. Yeah. So, and then the last one over here, this is going to be a little bit of a challenge, because I would actually like to use now a little bit of this here, of glycerin gelatin. And for this reason, I have to melt it, but not too much. So what I'm going to do is I'm going to simply put it here to 40. Okay, so it's also going to help, it's going to help it dry a little bit. Let's do it like this. And I'm going to simply go in here. I'm going to take out a small amount of this here. You didn't see it. This here should be enough. How am I going to do this? Can I get it on here? Here we go. It's very sticky. It's a little bit. I think 40 is already. It is way hotter than 40. Let's go down. I'm going to turn it off again. See, it's already melting here. It's already melting. Yeah. And now I'm going to add another cover glass on top here. Let's see if this is able to. Yep. How well this actually works. So here we go. Yeah. So that's it. So again, a short explanation of what I've done here. This here is glycerin gelatin. Okay, and the thing is that you see that it's pink because they've added a stain to it, because it's actually designed to stain pollen. Pollen are also very sensitive to shrinkage, and if you want to mount delicate water organisms, they generally recommend glycerin gelatin because it contains water, so it does not completely dry out. Yeah, actually, you also have to put a ring around it and so on. But what we're going to do now is we're going to now have a look on how well this actually worked. Okay, so we're going to first remove again this slide here. So that's the centrifuge one. So I'm going to go out of focus. I'm going to remove those two. And let's try now. I'm going to start again at the beginning. With the low magnification, we have to raise it up. And of course it's too. So let's see. Too bright. So here, this is 4 times objective. This is the one with this glue over here. Okay. And let's have a look. And of course, next week I would like to, of course, compare it as well. Let's go up with the magnification. Let's focus a little bit better. I think it looks kind of okay. And we have to open it. Okay. And it looks, I think, reasonably okay. So 60 times. I don't see a huge difference. So I think it might be actually okay. So that is now using the the Uhu, this glue over here as a mounting medium. Of course, the glue will now inhibit the movement of any organisms in here. Yeah. Yes. First of all, even though it's not completely dry yet, I mean, it's very viscous, so this will completely slow down the movement in any case. So I'm going to now move over to Elmer's PVA glue. Also not a huge difference, I would say. The Uhu does not really say what it is. I do suppose it's also PVA because what I read somewhere, this is a very common thing. And sometimes they don't disclose it because it's some kind of a trade secret. Generally for hobbyists, hobby use, I would simply try it out, right? If it works or not. And of course, for real reproducible scientific work, you don't want to use that because it's simply not standardized. But in any case, it's as you can see, it also seems to be working. But again, then they're not dry yet. And next week we're going to check if there's any bubble formation or if somehow the colors fade or if there are any other problems. Yeah. So, yep. Yeah. Yeah, so that's it says here, every small satellite will be brown over time since the chloroplasts will die. Yeah, not only that, they basically will start to bleach out. So we've got some very old microscope slides from the 1980s. And indeed, the chlorophyll, the green pigment, slowly starts to fade over time. Yeah. So now let's go over now to the glycerin gelatin. Okay, it's pretty dense. But it's already something that I do notice. It's already something that I do notice, and that is the contrast indeed seems to be much better. And this, I suppose, has to do something with the refractive index. I mean, the density is quite high here in any case. But if we just compare, what's this here? It's an interesting one. You just compare this here. Yeah, see, it's still liquid. There's still some movement going on here. Yeah. And I wonder if it is completely mixed. But if we compare this with this, I mean, honestly, yes, there is a difference. This one is not quite as this one is not quite as crisp as the other one. That is that's indeed it's still great, but it's not quite as crisp. So there is the difference in refractive index does seem to be visible. You've got a better contrast here. That's interesting. Yeah. A reddish green circles spotted. Could this be red? Yeah, why not? Some there are these what are they called? Hematococcus, maybe. Interesting. What is this? Look, did you see this? Where's this? Where's the arrow? I wonder what that is. Look, this is square. Square one. So yeah, so I think I'll wait a little bit. We'll wait until next week. We just ignore that the density is here much higher than the other one, but still, I do get the impression that's how comparison here also. Yeah, the glue is not as crisp and as high as in contrast as the glycerin gelatin. This is also the Uhu glue, the yellow one. Yeah, there's a comment here that like the gelatin didn't mix in. I did actually see it spread, but it could indeed be maybe that there was some kind of a this one here is definitely dry. So if it looks weird like this, then this is actually dried up. So it did not reach it here. You know what? I'm gonna try it again because it was a glycerin gelatin. It's something I actually would like to know a little bit more. So what I'm gonna do is I'm going to take again a small sample here, and now I'm actually going to try to dry it completely. I'm going to show. So I'm just gonna repeat this here. Yeah. So let's do the following. I'm spreading this here, and now I'm going to actually dry it. Okay, let's actually completely dry it because that's actually the way it should be. So we can see that because there's so many algae, luckily it breaks the surface tension. So this actually stays quite nicely apart here. I'm going to quickly warm it up, not too hot. It's not a point. I'm not trying to heat fix this. I'm just trying to speed up the drying process a little bit. It goes too hot too quickly. Turn it off again. Yeah, it's better. Not too hot. As long as the water is there, it's gonna be fine. So, and now I'm going to, yep, it's dry, I think. It looks pretty dry. So I'm going to now try again to add a little bit of this here, because now it should definitely mix in completely. Add another. That's a lot. That's a lot. It's not the most professional way of doing it. I ran out of toothpicks. So here we go. And I have to carefully melt it again. You know what I could try? I could actually try to, where is this? Yeah, here it is. Let's do this again here. So let's be patient. Usually, when it starts to melt, then it goes actually quite quickly. Did not start melting yet. So, a little bit. Now it sticks to the. Do another following. Here we go. So let's be patient. I mean, I don't want to heat it up too much. Maybe we should put it. Yeah. No. Is it going or not? Let me put on my glasses so that I can see better. Yep. Now it's slowly melting. Let's put it on here. And let capillary action do the rest. Here we go. Yes. And let's have a look. Let's put this away. And this goes away. And new slide. And I have to change, of course, the microscope view. Where is it? Here we are. So that's now using 60 times. And I have to tell you, I'm quite happy. Yeah. The jelly gives me Despicable 2 vibes. Yeah. Yeah. So Yeah, so basically, what I expect now over the next couple of hours is that some of the cells might actually absorb some of the dye and they might actually stain slightly red. Yeah, the pollen grains at least will do that. But I would be now interested a little bit in of course, in the stability of this, color stability and so on. Yeah. But as we can see that even though I dried it completely, the cells generally seem to be fairly intact and not shriveled up. Because I will tell you something honest, I tried to make some today in the afternoon. I spent quite a bit of time trying to make some nice permanently mounted slides of ciliates and rotifers, but they are extremely drying sensitive. So as soon as they start to dry up, they will shrivel up and completely change their shape. And the reason why I think that the algae don't do that, it's not only I think so, I'm quite certain, is because they have a cell wall, right? So their cells are simply much more structurally stable than those of animals. Yeah. So generally, I'm quite happy. Let's do the following, just regular bright field. Yeah, so that's basically the way that we should be able to see it, and yeah, looks surprisingly nice. Yeah. So in case you wonder, I mean, okay, occasionally there's some bubbles. But normally what I do is if I want to take a picture of this, of course I take a picture, but then I always usually do some contrast enhancement so that the background becomes brighter and the green starts to become a little bit darker green for better contrast and visibility. And that's usually something that I also do. Yeah, so if you want to have nice pictures, then you almost always do some contrast enhancement. Yeah. Yeah. So I'm quite happy with the result. Okay. And I think the next thing that I might be doing is maybe I think I'm gonna do a time lapse, a little bit of the movement of the algae and of the cyanobacteria. Yeah. I remember this was a long time ago, over 30 years ago, when I started to study at university. We of course also did some in an introductory biology course, yeah, some algae identification and things like that. And so nice. I was hoping for this pediastrum or or related. So I'm gonna show you. It's unfortunately a little bit covered up, but this is also a very, if isolated, they look quite nice. It's this one over here, right? Yeah. So unfortunately, I cannot go higher with the magnification because yeah, if I put oil on that, then it's yeah. So that's a pediastrum. Sometimes they unfortunately now covered up a little bit and a little bit incomplete, but they can be quite nice and very symmetrical. Volvox I've rarely seen. Yeah. And but you know what I would like to do now? Just it reminds me. Just give me a second. I've got a box here of. Let me see. Yes, 15. This here is a very old slide. I think from back from the 1980s or so. Where is this? Yes, this one over here. Let's have a look at this because it kind of shows a little bit how the colors fade over time. Yeah, I'll just show you the slide first. So this is a commercially prepared slide. Yeah. Most likely they also use glycerin gelatin because it's a ringed slide to kind of stabilize the cover glass and prevent moisture from entering or exiting, because glycerin gelatin, after all, is water-based. And this one over here is Spirogara, and it has also this typical chloroplast. But let's have a look at at how if over the last 30 years or so, if the color faded. Okay, so let's have a look here. So I put it into the microscope now. So that's still the mine. And now let's have a look here. If I'm only able to find it. Maybe that's one of them. Nope. Let me quickly have a look here. And otherwise, no, you know what, I'm just going to try this at a lower magnification. I just have to find it. Here we go. Ah, yeah, here we go. Okay, that's it. Okay. So this is, yeah. So very nice. It actually shows, do you see those, the spiral-shaped chloroplast here, right? That's supposed to be green. And it's not green anymore, obviously, because, yeah, over the years, the color has faded. Yeah. Or basically over here, it's completely black-white now. Yeah. But the green chloroplast in the center, now my mouse is acting up. Here, this green chloroplast in the center is not green anymore, right? So that's one of the disadvantages of permanently mounted slides. That yeah, obviously the chemical is not stable. Yeah. Is this unstained? Yes. Basically, algae and pond water samples you rarely stain because you actually want to see the green chloroplasts. So there is actually no need to stain them. But in this old slide here, as you can see, that basically they lost the natural color over the years. It kind of bleached out, so to say. Yeah, because of the light of the microscope and so on. This kind of all bleaches it out. Yeah. So I'm just saying that this is something to be a little bit expected. Yeah. So, but let's go back again to the one that I had before. So that's the one that I just prepared, and you can see that it's still, the algae is still green. Yeah. So let's do the following. Before, I'm going to do now something extreme, okay, something which you should not do, but I still want to demonstrate this. Because after all, we all want to learn something here, including myself. I'm now going to use a completely unsuitable mounting medium, which is Euporol, which I used for my insect wings. Okay, and then what time is it? Yeah, that's the last thing that I'd like to do, and then I'm gonna move over and I'm gonna recap a little bit the insect wings. So what I'm going to do again is the following. I'm going to, where is the desk? Here's the desk view. So let's add again a small drop of algae on here. Let's spread it, and now it's really important because Euprol is hydrophobic. This has to be completely dry. Not a problem. We're just going to dry it, and either air dried, or we're just going to again use the coffee warmer. Okay, and then I'm going to use Euprol mounting medium, which contains a solvent. And this is, I'm going to show you the problem with that, right? Why it's so important to use water-based mounting media. And I don't want to fry it too much, really. Okay, so it's drying. Do chloroplasts contain DNA? Short answer, yes. Chloroplasts contain DNA, and mitochondria also contain DNA. Having said that, it's fairly small compared to the rest of the DNA, but scientists actually have done DNA sequencing of chloroplast DNA, and not surprisingly, the DNA of chloroplasts is very similar to those of cyanobacteria. Okay. What would, if we were to mix some of the algae with a diluted PVA, why not? We can try that. I have not tried that yet, but I see no reason why it should not work. Okay. What will it look like if you cook it too much? I don't know. I can also try that. But usually what you do is it's almost a little bit like heat fixing of bacteria. I suppose maybe it's not gonna happen. Not a lot is gonna happen because the cell walls are gonna be stable anyway, right? So this is basically now the one that I would like to test with Euprol mounting medium, and this is a completely unsuitable mounting medium, and I hope that I'm able to demonstrate this. So I've got another cover glass here. It's an unsuitable mounting medium because it contains a solvent, and the solvent would actually dissolve the green pigment. Look, the solvent here also dissolves the this here. Just so that you see how. It's okay. So let's try this. Am I able to get? You see that it doesn't close it off again. You see that's a problem. It doesn't close it off. Let's try it again. So let's go down. Here we go. It's not a lot, but but enough. I have to completely exchange this here. Look at this. That's so, and now this goes on top. Let capillary action do its work again. Maybe you can press down a little bit to help it spread a little bit more. And let's now put this under the microscope. And I need to switch over again to the scope view. So this is the one with the glycerin gelatin, and this over here is now the one, with Upril. And you already see that it looks a little bit paler than the other one. Okay, let's go up 60 times. Okay. And the reason is, and I hope to be able to demonstrate this, and maybe we're able to see this next week better. What's going to happen is that the green color, the green pigment, is actually going to leach out of the cells, and they're going to lose their pigment because the solvent dissolves it. Yeah. So then basically, it will look much less contrasty. So that is the one with Upril, and over here again, that's the one with glycerin gelatin. And I think you see that there's a huge difference already. And this one looks way flatter and less contrasty. So the question is, sustaining it for long-term color is good. Depends on the specimen, but indeed, this is for certain histological specimens, they do stain it, and it's then visible. At least the structures that you want to see are visible for a very long time. But even those stains sometimes are not always entirely stable. Yeah. So glycerin gelatin, you need that if you want to work with that. I tell you, you can try to make it yourself, but it's really difficult to make yourself, because you also have to add a disinfectant. Okay, because otherwise it's going to turn moldy. So that's a little bit the problem. And what I almost recommend is if you want to get it, you can actually buy it. I mean, I got this from Brunel Microscopy, and this one is the one that's the red one. Basically, is the one that already contains the stain. It's suitable for pollen, but there are different ones also for other water organisms. Yeah. But strictly speaking, if you really want to use this, and if you really want to have a long-term stability, you strictly speaking have to add a ring around it like this. Yeah. You have to, because this seals the cover glass, because this will not completely dry out, and it should not dry out. And in order to stabilize the whole thing, and also to prevent it from turning moldy and bad, and from absorbing moisture and giving it off again, depending on the humidity, you have to actually, strictly speaking, put a ring around it if you want to make sure that this stores for long term. Alternatively, and I know this is a little bit uncool if I say that, alternatively, especially with algae and water organisms, also where movement is so important, maybe not make permanent slides, but simply keep on observing them and documenting them using videos. I mean, that's what I'm trying to do now as well, because sometimes movement and so on is quite important. Can you identify the genus? Well, using the book, sometimes you can. But then the biodiversity usually is so high that even if there's something in the book identified all the way down to the species, chances are pretty good it's not the same species. Yeah. Will alcohol bleach them? Yes, definitely. And not only that, alcohol will also shrink them. And that's actually the problem with Elmer's PVA, because there's a solvent in there. Not Elmer's PVA, Upril. Yeah, because there's a solvent in there. And Elmer's PVA, and the other one that's actually water-based glue. Yeah. Yes, I knew it once. It is an antifungal. You can actually get the recipes online, but I know that on Microscopy UK, I've once read an article that somebody made glycerin gelatin at home and added Listerine mouthwash as a disinfectant. Listerine mouthwash, a couple of drops, and this also preserved it. Yeah, but I said, no, I know it's called the antifungal substance called thymol. Thymol. Thymol is an antifungal substance that is added to glycerin gelatin. Not phenol, thymol. Yes, yes. I think it's yeah. How about vinegar? You gotta be careful with the pH because sometimes if the pH is not correct, like you're adding vinegar or so, you're changing the pH, and this might actually change also the colors because some natural colors are pH sensitive, and modifying the pH might therefore be a problem. Yeah. So I tell you the following. I bought Brunel. I'm in Austria, right, in Central Europe, and I ordered this from Brunel, I don't know, 2 or 3 years ago. They just sent it without problems. So I will tell you what they did not send. They do not send any liquids. So when you buy stains, when I bought stains from them, I got them in powdered form. And also, they did not send any liquids for ringing, for example, the slides because of legal issues. But so, if you want to buy some stains, I bought some stains. I got a little bottle like this with a small amount of powder. Yeah. So at least at that time, at least 2 3 years ago, I did not have any problems. Yeah. Yeah. So that is, and yeah, vinegar and gelatin. That's the pH that's always a sensitive issue. Yeah. So just saying, you have to experiment here. Yeah. Okay, here's some info. They will send solids globally. You can always add the liquid yourself after, and they will advise it to use it if you need if it needs a more aggressive solvent. Yep. So that's a little bit unfortunate. So it's I remember a couple of years ago, I could also buy some other solvents, or even over Amazon, some immersion oils and so on, and not anymore. Yeah. Yeah. So having said that, what time is it? Okay, I want to very quickly change topics and simply give you a very quick recap of last week's. That we quickly have a look at last week's slides, which are prepared. Okay, so yeah, so that's one. That's the other one over here. Let's put them away. And I would like to show you. Let's have a very quick look at the insect wings, and then I would like to say thank you very much to someone who sent me some pictures as well. Just a second. Yeah. Christoph, he sent me some pictures of some insect wings. Very much thank you. Where can I find this? Where are they? I think it's here. Nope, this was the birthday cake. This was the one over here. The slideshow. Because I made videos, so these are basically UV. That's using fluorescence microscopy of insect wings, and first in bright field and then using UV light. And yeah, and I want to say thank you very much for having sent them to me. I got the permission to share them with you. And I just want to show you again. Let's go. Where is this? Where's the slideshow? Let's go to the very beginning. Okay, so these are wings here with all of the technical detail. First in bright field, and this one is now using UV light. Okay, and you see that. This is okay. You get the idea. Okay, this is yeah. This is basically using UV light again in bright field, and next one here, yeah, using UV light, and you see that using UV light, part of the wing, insect wings, start to light fluoresce, yeah, and this is quite a nice thing. Of course, fluorescent microscopy is not something that everyone has. I don't have it either. Yeah, but it's quite interesting to see that. Yeah, looks pretty. Yeah, and yeah, here as well in insect wing. First on the regular bright field, and over here, yeah, using UV light, ultraviolet light. So thank you very much for sharing this. But this actually needs it needs a dedicated microscope. Yeah. So very nice pictures. Yeah, thank you very much um, for sharing those. I really like them. Yeah. So thank you again for Christoph. I don't know if you've joined in now and if you're also able to comment on that, but if you want to, okay. I would like to, of course, invite you or either right now in the chat or later on in the comment section below. If people have maybe questions, yeah. Yeah. Really nice. It's yeah, it's a Leica microscope. Yeah. Okay. Ah, interesting. There's an issue with a 404 knot. I didn't have any problems really. And I was also with Brunel. I was also in email contact. So basically, there was an issue because they could not send some of the chemicals because of legal reasons. They asked if something, yeah, if they can send a different one. So there's actually, how do you say, there are actually people there that you can talk to, which is quite nice. Yeah. Yeah. So thank you very much again. Also very nice pictures. And now back to the microscope view. Let's have a very quick look at some of the some of the slides that we prepared last week from the insects. Also, I do not want to spend too much time on this, but let's see a little bit how well this works. So I have to find the wing. Here it is. Yeah. Yeah, you see. I allowed it to dry now for a week, and we can see that it's still, of course, quite stable. Yeah. Especially when using mounting media that are hydrophobic or resin based, not water based. It's really important that there is no water in the specimen. Otherwise, what's going to happen is that there's going to be water enclosed under in the slide, and this can actually make it very unstable because bacteria and so on can start to decompose it, because the water is not able to escape. Right. For this reason, you have to make sure that if you use UPRL or a similar mounting media, you have to really make sure that they're completely dry and free of water. So this is the one that was a little bit twisted around, but you can actually see that over time. Yeah, the structures also do become a little bit brighter. Yeah. So looks quite nice. Yeah. So okay, so there seems to be a technical issue with Brunel. Okay. So let's have another look. I mean, I do have, of course, I do have a flashlight, a UV flashlight, but this is, of course, not neither strong enough to do proper UV microscopy, fluorescent microscopy. Yeah. So let me check this one over here. Yeah, also quite nice. Especially when doing fluorescent microscopy, what you sometimes the mounting medium itself also has something called autofluorescence. So usually for fluorescent microscopy, you also have to make sure that you're choosing the correct mounting medium so that the contrast is best. Yeah. Yeah. So that is the question of Marie Chardon. Hi all, I wonder if you can shine UV light on the specimen and then take a picture, and the answer is yes and no. There is something called blue light excitation. This basically means when you use a microscope with a halogen lamp, which contains also not only a lot of red, but also a lot of blue light. And if you use a very dark blue filter, and if you have a very long time exposure, you're actually able to see that the chloroplasts start to fluoresce in red. But this is a very weak signal, and yeah, it's called blue light excitation, and it kind of goes a little bit into fluorescent microscopy, but it's not so, the contrast and the image quality is by far not as good. I mean, I've also got some blue light LEDs here, but I was not able, you won't be able to see it, it's blue, and I've got also UV LEDs, but I was not able to get the same effect. Yeah, it's not strong enough. Yeah. And here another wing. Again, I mean, if you've seen last week's live stream, I've thrown this together quite quickly. So this is folded up and not so nice. It was more a proof of concept. Yeah. Yeah. Okay. I'm just reading a little bit here. Okay. Okay. Christoph is here. The base is a Leica DM IRB inverted scope. It's an old research scope. I use a regular HPX light source as well as laser sources and a 5 times to 150 times flutar lenses. That's the next thing that I don't know if what your setup is, but sometimes what you have is you have actually light UV light actually shining from the top through the lens, through the optics onto the specimen. So sometimes there is it's at least this is used in some so-called material sciences microscopes. They sometimes have a separate light path which goes through the objectives. I wonder if this is the same case here as well. Yeah. Okay. Okay. Still a little bit of an unfortunate thing here that the web page of Brunel does not always seem to be working. But then I recommend maybe to send them an email. Because yeah, at least some time ago, I mean, there was absolutely no problem in being in email contact with them. I bought already several things from them, including also a slide ringing table. Yeah, here. This one is I remember this one is from the ladybug. Yeah, the front wing. And these are the dark spots. Yeah, on the red front wing. Yeah. So yeah, I'm just reading again some of the comments here. Okay. Yeah, so that's a little bit the thing. So generally, I've found it to be fairly stable, though the UPRO and the I'm also with Elmer's PVA was also quite stable, but essentially, I think it depends also a lot on yeah, how much water is present as well, because some of the specimens that I made long time ago, they started to decompose and so on, started to turn bad after a couple of years. Yeah. Okay, that's interesting. So there is indeed a problem with their web page, which is kind of unfortunate. And let me quickly, for the last, what time is it? Oh, I'm just gonna quit anyway in a few minutes. So let me quickly check here if this here is still okay. So that seems to be still the yep. Okay, that's the one from before again. I guess I quickly have to label this so that I don't forget which one's which. Yeah. Not that was not the right one. See, I'm already getting confused here. This is the one that I wanted to do. Okay, let's see here. Yeah, that's the one. And this one over here. Yeah, that's the one with the UPROL, I think. Yeah, and that's the one which is going to probably lose its pigment over the next couple of hours. And it's going to start to become brighter and brighter as the chlorophyll is washed out by the solvent. Yeah. So yeah, now we have to wait. And that's the one with glycerin gelatin. Yeah, I'm quickly going to do the following. I almost cannot believe it myself. Let me quickly check myself a little bit the whole thing here. Brunel microscopes. Really, how is this possible? I mean, the website is still on, is still active here. Secure shop. Okay. It's interesting. Okay. Mountains, slides, cover slips and mountains, solvents and reagents, acid alcohol. I mean, you got everything that you actually want. Isopropyl alcohol, sodium hydroxide. I mean, at least I'm able. I mean, I don't know where you got the 404 error, but with me, maybe I don't know if you go to checkout, but at least with me, it seems to work quite fine. Okay. Maybe I mean, I didn't click all the way to the end, so yeah. Okay, you ordered it. Cool. Yeah. If you order glycerin gelatin, just be aware. Maybe a last warning. Last warning, because something that you have to be really careful that you don't do. You're not allowed to heat it too much. Okay. Let me quickly show you the desk here. So you already know my coffee warmer here. Generally, there is a saying it should not be hotter than 40 degrees. However, this one is definitely hotter than 40 degrees Celsius, right? Because what happens is if you heat it up too much, then it will not solidify anymore. So you can actually break the glycerin gelatin once you heat it up too much. In the past, I used to always heat up the whole bottle, either on a coffee warmer or maybe you can use a warm water bath. But this takes a long time. So for this reason, I've changed the system around, and I always have taken using a spatula. As you've just seen before, I've always taken a small amount out, and because this kind of protects the original. Because that's the thing. You always want to close it again to prevent any spores and whatever falling in here. And if you actually smell it, it does smell like phenol or thymol or whatever. It has this typical hospital disinfectant smell. Yeah, and that is because they added this disinfectant. Otherwise, it's going to turn bad. Yeah, just saying in case you're wondering. Yeah. So concerning the coffee warmer, it's good for coffee as well. But there are many coffee warmers can be plugged into a USB port for electricity, but the USB ports are not able to provide a lot of power, right? So that's why I used just one that can be connected to the mains. Yeah. So this is actually quite useful, especially also if you want to make crystals under the microscope for microscopy. For drying the crystal solution, it's quite convenient. Yeah, but don't trust the temperature. Okay, so what I would like to, I'm just quickly going to go through here again. Okay, the 404 was when trying to add to the cart, but it was an old site. Oh, really? Okay. Okay. Yeah. Okay, I got it. So now I got it. It was actually the old on the old website. Yeah. So yeah, I mean, I would be happy if you buy something from them and did some experimentation. You report back as well. Okay, I have been buying already several stains from them, and was quite happy with the results. From my side, I think I'm gonna call it quits for today again. Just a second, there's a comment here. I've just acquired Canada balsam. What should it not be used for? Oh, Canada balsam. Okay, Canada balsam is a traditional mounting medium. I'm just gonna, because there's a question, I'm just gonna answer this here as well. Is a traditional mounting medium, and it has the advantage apparently that if done properly, it apparently stores indefinitely. The disadvantage is, however, that the specimen has to be completely water-free. This basically means you have to dehydrate the specimen, first in alcohol, then in isopropyl alcohol, and then afterwards you have to put it into solvent like, for example, xylene, which is not good if you inhale it. But it has to be completely water-free, and because it's also solvent-based, it's not suitable for any specimens that have pigments that dissolve. So similar to, for example, Uperol, which is also not suitable for those specimens that basically have pigments that can dissolve. My suggestion is experiment with it. Okay. But yeah, Canada balsam is a traditional mounting medium that has been used in a quite wide variety of different settings. But the specimen must be completely dry. Yeah. Someone mentioned earlier that the warmer is more than 40 degrees because it's supposed to warm up coffee with up to 40 degrees. So maybe the cup with a boiler. That could be the possibility that if you put a cup on top of it, and also there is also this rubber, how do you say, surface on here, which also of course takes some of the temperature away. Yeah. So ideally you should always measure the temperature separately with a thermometer. Yeah. So I'm gonna call it quits today. Okay. And I hope that you liked it. I wish you all the best. Okay. I'll try to make a couple of more videos in the last couple of months, weeks and months. I've not made so many videos on this channel. also because I was quite busy with school as well. But have a nice weekend, and yeah, hope to see you again. All the best. Bye bye.